Yes. Hello. Yeah. I can keep this off now, right? Uh,
Hey, good afternoon, everyone, and It's a pleasure to have Roni give the second talk on the Nobel Prizes of this year. This one is on the Nobel in Physiology and Medicine, on microRNA and C. elegans and how it revolutionized biology. So Roni, as you know, joined here in 2020, I think, after BS at IISc Bangalore. And he's working with mainly Sandeep and Vani. And yeah, go.
Yeah. Thank you for giving me the opportunity to present this talk. So today I'll be talking about the recent Nobel Prize in Physiology and Medicine. The title of the talk is Early Birds and the Worm: How Discovery of microRNA in C. elegans Revolutionized Biology. So before going into the talk, I will go over some terms which will come up like multiple times during the talk. And most importantly, there are these two terms which I want you to remember: transcription and translation. So when I'm saying transcription, what it basically means is there is a DNA sequence, and from the DNA sequence, you are making a RNA sequence. And then the another term is translation. So what translation means is that you take the messenger RNA sequence, and from that messenger RNA sequence, you synthesize a protein. Okay, so these two processes are very key for the survival of an organism. So a bit of introduction to this year's awardees. So this year, Victor Ambrose and Gary Ruvkun won the award, and both of them trace their lineage to Sidney Brenner. So Sidney Brenner is the person who started the entire C. elegans biology field. He established the organism as a model organism. He actually used to, go out and collect multiple C. elegans like species and then try to characterize them in the lab. And Brenner and H. Robert Horwitz and Sulston, the three of them were also awarded the Nobel for establishment of C. elegans as a model organism and other works as well. So turns out that both of them were postdoc in Robert Horwitz's lab. That's where they met. And from then onwards, they established labs of their own. But most of the work that they have done, which won them the Nobel, was done in extremely close collaboration with each other. So, yes. Okay, fine. So, like generally, when we talk about RNAs, the most canonical one which we know about is the mRNA or the messenger RNA, which we know. But turns out that messenger RNAs are they are a significant portion, but they are not the only RNA inside your nucleus or inside the cell. You have ribosomal RNAs. So the ribosome is the complex which is synthesizing the proteins, and turns out that majority of its enzymatic activity and shape structure comes from RNA themselves. And the ribosomal RNAs work with something called transfer RNAs, something called a tRNA. So these two are for protein synthesis. And there are other forms of RNA, which like your microRNA, which are more geared towards regulation. So there is a whole class of regulatory RNAs, which are like long non-coding, short non-coding, repeat RNAs, and the more and more these sequencing methods develop and become more and more sensitive, you come up with newer and newer variants of RNA. So RNAs in general are single-stranded. You can have double-stranded RNAs because the complementary base pairing is obviously there. And turns out that RNAs are synthesized in a single-stranded way, but generally like they form some sort of secondary structure. So if you look at tRNA, for example, it has a after it's synthesized, it will adopt something of a hairpin loop structure, like something like this. It looks like, if I'm not wrong, and then like there are four. Yeah, it has something like this four looped like clover leaf kind of a thing. So all of these come with their own secondary structures, and turns out that this thing can fold in the sense like this folds onto this goes like this, and it becomes an L-shaped thing. And all tRNAs do this. So there is some structural information encoded, some sequence information encoded. Yes. So the presence of these RNAs were widely known. Like ribosomal RNA and tRNAs were known by the time they were, carrying on their research. So
all RNA have the same code base pair.
yes, AUGC. Yeah. Instead of thymine, you have something called uracil that becomes U. Okay. There is no thymine in RNA. That the sugar molecule is ribose instead of deoxyribose. These are the only two differences from your standard DNA. Okay. So these are the early birds, people who did the foundational work, and this is the worm. So this is our little model organism, something called Cenorhabditis elegans, C. elegans. It's a roundworm, near about 1 millimeter long. And the interesting feature for which Sidney Brenner chose this as an experimental organism is that its shell, the egg shell, is transparent. So if you look at it under a microscope, you can see the divisions clearly. You can so it turns out that there is a very nice degree of order in this thing, in the sense that every cell division across all C. elegans species will have the same pattern if it's not mutant. So all white type cells will have a single cell. It will divide slightly asymmetrically, like this one will be larger, and then there will be a cleavage at a slightly oblique angle, and then everything is set in stone. How cells will divide, how cells will choose their ending fate, like whether to become skin, whether to become neuron or whatever. So everything is fixed from the moment the first cell division has occurred. So this is the life cycle. They have very short life cycles, near about a month, and they're very easy to grow and care for in the lab. So they come in two variants actually. So they are you can either have hermaphrodites, which are like male female combined, right? And then you can have like male and female separate. I mean a male separately. So a male and hermaphrodite can mate, or the hermaphrodite can self-fertilize and give rise to progeny. So these things, they lay eggs. The eggs then go through four larval cycles before becoming an adult. Depending on how much nutrition they get at this stage, if they get less nutrition, they can go into a arrested state called a dower. And if you provide them nutrition, they again like directly jump to L4. So this is something like a spore kind of a thing you will have in fungi. So they have some access to that as well. So these are like the external characteristics, but going more towards the cellular things. So, okay, I have skipped a slide, I think. Okay, okay, I will come back to that later maybe. So another interesting thing about this organism is that it has a fixed number of somatic cells. So what does somatic cells mean? Somatic cells means that non-reproductive cells. So every hermaphroditic C. elegans will have exactly 959 cells. If it's wild type, exactly 959. You can map the cell fate of every single cell. Okay, that map is known. At the end of the growth, yes. And you can tell that all the progenies of this cell will make these cells. The entire fate map is like fully known, completely known. Okay, so now what was known at the time of Rafkun and Ambrose is that people knew about general gene structure. So in general, let's consider that this is a DNA. A DNA will have something called a promoter here, and there will be some regions upstream of the promoter, away from the promoter, where something called a transcription factor can bind. So these transcription factors are basically proteins which come and bind to the DNA at specific sequences depending on specific cues. So let's say you have some sort of heavy heat outside. So some transcriptional factors will activate, which create proteins which you need for like heat resistance and stuff like that. So these transcription factors can be of two types. You can have an activator, which when bound can initiate strong expression of a gene. You can have a repressor, which when bound completely turns off the gene or reduces the expression by a very large extent. And when it is when nothing is bound, it can either be off or have slightly weak expression. This thing is basically because since the RNA polymerase binding is stochastic, you can still have some degree of expression in these, even in transcription factor unbound states. So interesting thing here is that the entire regulation is happening via proteins, right? Transcription factors are proteins. You have other signaling receptors sitting at the surface. You have other signaling molecules at the surface, which are also proteins, and they're sending the signal to the transcription factors, activating, deactivating them and stuff. So gene regulation via transcription factors was well known and was beginning to be well understood. People had figured out some machinery and stuff by that point. The surprise and why is gene regulation required? So imagine this: all cells in our body contain the same genome. They contain the same information encoded in the DNA, but depending on which transcription factors are active in each cell type, you have different sort of cells. You can have a neuron, you can have some muscle cells, some intestinal cells, some fat tissue, whatever, right? So if you have any sort of dysregulation in this process, you end up with tumors, cancer, and all these associated whatever diseases, right? So what do transcription factors do? So transcription factors are now binding. You have transcription giving you some mRNA, and the mRNA is then now sent into the cytosol. So mRNA is made in the nucleus, but it is now sent into the cytoplasm where the ribosome is gonna create proteins via translation.
So each mRNA produces one protein.
No. So okay. So you have a DNA sequence, right? And you have a promoter here. Depending on how many cycles of RNA polymerase activity has gone here, you can have multiple mRNA molecules. Similarly, when you have an mRNA here, and there is another ribosome going as a motor on this thing, like not a motor, I mean, it's like walking on this, you can have multiple copies of protein from the same mRNA. So it's like amplifying in some sense, like from one DNA sequence, you can create n number of proteins. If
But
by
one single mRNA,
works
for only one protein.
One single mRNA will yes, once fully processed, a fully processed mRNA inside the cytosol will produce only one type of protein. Yes. I misunderstood the question. But again, from the same gene, you can have multiple different mRNAs because of splicing and all this business, which we'll not go into. Okay.
Or it will get destroyed?
It has a half-life. So mRNAs are degraded because, again, like once the signal has gone away, you don't want the protein to persist. So mRNAs, all these nucleic acids, right? DNA, mRNA, RNA, whatever, they have their associated nucleases. So they will cleave them off over time.
What is the time scale? Like, I don't know if it's a valid question. So
how
many proteins might be?
So that's a slightly tricky question to answer, because what can happen is the half-life is contingent on multiple modifications on the mRNA itself. And depending on the length of the mRNA, again, like the amount of time the ribosome is taking to walk, you can imagine, since ribosome has a limited pool, the total number of proteins it can output also will vary. So there is multiple modes of competition in the system inbuilt, which also like slightly regulates the system. So there is some sort of feedback. Okay.
But it is all subject to control?
Yeah. There is control built into everything. Like,
and that can
very
be modified depending on the need. Then
yes.
Right. Okay.
So this is where the microRNA things will come in. So transcription factors regulating DNA gene expression was pretty well understood. But then spuriously, people were doing some experiments and they were seeing some strange things. Like, let's say you take something like RNA injected into some cell, like just single-stranded RNA injected into some cell, you will see that the expression of some genes will go down, right? Another thing is like you can inject double-stranded RNA as well into cells, and you will see that their expression levels are going down. You see this reduction in expression in plants. If you inject mRNA, if you inject RNA, you see it in C. elegans. So all of this phenomena which was being observed were like brought into the umbrella of a term called RNA interference. Okay, so this is where the story begins. So RNA interference was like characterized and described in like good detail by Andy Fire and Craig D. Mello. So I think Mello is from Victor Ambros' lab. Yeah. So basically they have lots of connections going on here as well. So Craig Mello, so they were able to like identify RNA interference, and then they were able to show that it's mostly if you inject double-stranded RNA that you get the most effect out of this RNA interference. Also, they found out that the RNA interference is catalytic in nature. So let's say you have injected one molecule of double-stranded RNA, it can inhibit like at much higher stoichiometric amounts the amount of protein expression. So there is some degree of catalytic activity as well. Okay, so this is where both Ambrose and Rafkun come in. So these people have identified, like people were studying some sort of RNA interference, and this is where our heroes come in with C. elegans, and they come in with these specific mutants of C. elegans, something called Lin4 and something called a Lin14. So these are mutant animals, mutant in the sense that some gene has been, like these genes have been changed. So phenotypically, what you observe, phenotypically means like in the organism's morphology, cell types and all. So this one, the Lin4 mutant, and you can see that it has these small bead-like things. So these are actually eggs. So this is hermaphrodite worm, and it can't lay the eggs because the entire vulva and the like midsection machinery which is required for the eggs to be laid does not exist in this worm. Okay, so it keeps on producing eggs, but it can't lay them, and so it has a bad time. On the other hand, this Lin14, what it does is that it completely forgets that it has to make these adult type of tissues. It only keeps making larval tissues. So these are something called heterochronic mutants. Heterochronic in the sense that they are locked into a state of development which does not match what should be at that stage. So these worms, immediately at larval stages, they will start producing eggs, which should happen only in the adult. These worms, even in the adult stages, they will keep producing larval tissue, which they shouldn't. So these two mutants were of interest to both Rafkun and Ambrose because they were characterizing the genomic loci of these mutants, and they were also able to figure out that like this one regulates this one.
The mutation, was it an accidental discovery or it was intended?
So these things are the result of something called a forward genetic screen. So basically the idea is that you give these animals some sort of a mutagen, radiation, some chemical, and you screen them based on some criteria you have. And then you find what are the mutants you have. And then like, so nowadays, what you can do is you can sequence things for very cheap, right? Finding the location of your specific mutation is very easy. Comparatively, but here they had to like clone and do a whole lot of like molecular biology work to figure out that okay, this is the Lin4 sequence and this is the Lin14 sequence. So that was the backbreaking work that went into this. Okay, so now they sort of know that Lin4 represses Lin14, so that was also known. So here, this is actually what these things are showing is that as I was telling you earlier, the cell lineage map is fully understood. So this is saying that some cell here, so this is inside the egg, you had that oblique cell division. So, and then you have these cell types, they will give rise to these specific cell types down the line. And then each of these cells, the fate of the progeny is also mapped, right? So now what, from this, what they were able to do is compare between different mutants of Lin4 and Lin14. So here what this is saying that this is when the eggs are hatching. At the first larval stage, this cell here is expressing your has specific amounts of some protein, which gives rise to specific cell types. But in the Lin4 mutant, the shift to this blue color never happens. So it keeps repeating what the level, like the first larval cycle would have been. In the Lin14 loss of function mutant, LF, it keeps repeating what the larval 2 stage should have been. And the gain of function mutant, it essentially behaves like this. And in the loss of function mutant, along with Lin4 mutant, also like mimics this. So what gain of function means is that, let's say I have a gene which is under the control of some other factor. Gain of function means now it is independent. It will always be expressed, whether the other gene is on or off. Loss of function means even if the gene is being expressed, it doesn't create any product. So the benefit of having loss of function or gain of function mutants is that you can like nicely chart out the biochemical pathway, like which protein comes upstream of which other protein, right? Okay. So this is some of the like early papers which were published. I'm summarizing them in summarizing the key portions. So what what Ambrose went on to do is he chose Lin4 as his gene of interest. Rafkun, on the other hand, went and worked with Lin14, and Rafkun was able to clone Lin14, which is a basically it's a nuclear, it's localized in the nucleus during the first larval stage and then gradually gets degraded in the next few larval stages. So, and then they were able to show that the gain of function mutants which they had so this N335 and N536. So these two variants were the gain of function mutants. Gain of function again means that this variant of Lin14 does not care whether Lin4 is there or not. It will always be expressed and it will always give you some phenotype, in the sense that like all you will have all the eggs, right? So so they saw that both of these mutations are present at the 3' end of the mRNA, which is synthesized. So that was interesting. On the other hand, Victor Ambrose in his lab, while sequencing this, trying to clone this Lin4, ran into the problem of Lin4 being such a small gene that it is very difficult to clone it. So identifying that gene took a fair amount of time. That is why all this mRNA identification is happening in 1991, but the characterization of Lin4 is happening in 93. So they are running a bit late in the timeline. So what they were able to show now is that this is a very small gene. So the immediate assumption was that this thing cannot be synthesizing proteins. As to have a protein, you need the mRNA, the structure of the mRNA goes something like this. You have the 5' end, you have the 3' end, and here you will have some untranslated region. At the 3' end, there is some untranslated region plus a long polyadenyl tail, right? And no way this small 100 nucleotide long Lin4 sequence can give rise to something as long as what is required for an mRNA. So fine, what they did is they tried to figure out like, okay, I have this gene, what is the product? So they were able to figure out that this gives you a product of RNA, which is there is a long form and there is a short form. The long form is near about 70 nucleotides. The short form is 22 nucleotides. And then like what they did was they were sharing their results, and on the fateful 11th of June 1992, they had all the sequences with them. So Rafkun and Ambrose like sit together and they compare the sequences, and they were able to find that if you take the shorter Lin4S, this Lin4 short sequence, you can find that it has some complementary portions in this 3' region, and the complementary portions are absent in these two gain of function mutants. So they had very strong conviction to say that this RNA, which they now are classifying as microRNA because it's short length, 22 nucleotide long RNA, has some sort of partial binding to your 3' untranslated regions. And it is this binding which gives you the transcriptional, which gives you regulation of the Lin14 amounts. So this is repressive in nature. So that's their hypothesis. And then they were able to do some bioinformatic analysis, run some more experiments where they overexpress Lin4 and see the amount of, yes.
So, what is the you have that picture there, something blue and then that box.
Oh, this one?
Yeah.
Oh, okay. So the blue one here is the mRNA sequence, single stranded mRNA sequence. Your this Lin4 creates also a single stranded RNA sequence, which has this hairpin-like structure. This hairpin-like structure now gets processed by some other machinery, which they did not identify. This gets processed into this, right? And this sequence has some complementarity to your mRNA, Lin14 mRNA sequence. So imagine
the middle arch is something produced by somebody else.
Which one?
The middle arch.
Oh, the middle arch is just because the complementarity is at two portions.
Correct.
And the middle portion has no complementarity. It is just like bulging out so that the other, it's just trying to maximize the amount of strong contacts it can make. So it has sort of extruded this.
So that's a microRNA.
The red one is the microRNA, which is binding to your Lin14 mRNA, right? And so here you have something of the sort of a double-stranded kind of a thing with some hairpin loop kind of. So the thing is, like, if you sort of were to imagine that you have some ribosome which is sitting and walking on an mRNA, you would assume that the regulation should be more at the 5' end from where it is starting to walk, right? Because then if the ribosome is seeing a double track, it will sort of get stuck there. But turns out that the regulation is happening at the end, like the ribosome can already walk through and then the protein is already made. Why does it care whether there is a roadblock here? So turns out that what actually happens is a bit more complicated. It's not that it's just is a sort of a traffic jam for the ribosome to walk through. If it binds here, there are other protein complexes which sort of inhibit ribosome from walking through. So I'll come to that as well.
Is this the first evidence of an mRNA binding to another mRNA? Is this the first evidence?
This is the first time when you are having, this is not the first time when you are saying that there is double-stranded RNA.
Okay.
That RNA interference people, they had already looked at something called small interfering RNAs, which are also double-stranded RNAs, but they are fully base pairing. And they don't base pair specifically at the 3' end. They can base pair wherever you want, right? So the RNA interference people who are working in parallel also have identified structures of this nature, but it just doesn't have the hairpin.
RNA came later. I mean, siRNA came later.
Those are like classification names, but so here in the timeline, if you see like this, they have like small complementary RNAs can hybridize. So hybridization is already they are seeing double-stranded RNAs, and this is 1983-84.
Okay, but the fact that they can play a regulatory role came in 98 or something.
Yeah. So gradually, like Mello in his Nobel lecture, which was at 2006. So he mentions that they like the people working in the RNA interference field, they had a hunch that these things are all using sort of similar machinery for their activity, but to conclusively prove that these are two different mechanisms, siRNA and microRNA, and what machinery they are using, it took a bit of time. So like here, like things are a bit, what you would call muddled, like classifications sort of lose some meaning. Yeah, they're running in parallel tracks.
How stable are these mRNA?
So microRNAs stable in the sense that these are also degraded depending on your external cues. So, but the time scale of the stability, I'm not exactly certain. Like whether they are of the same scale as your mRNA or they decay faster, slower. I'm not exactly sure of that. I'll have to take a look.
And all they are always repressing in nature,
Not always.
not always.
Also, they don't always bind at 5 prime. I mean, those are like exception things which I'll like show in one slide, but
But the
yeah.
mechanism is the mechanism similar to how transcription factors regulate
no, no. Transcription
the gene
factors will go
expression.
and bind
Yeah.
at the DNA. So that is at like inside the nucleus. Here, transcription has already happened. You have something called post-transcriptional control.
No, I'm talking about the binding mechanisms, like binding, unbinding, like
No.
how the
Because the transcription factors are amino acid nucleotide interactions.
Okay.
Here you have nucleotide interactions. So this is mostly base pair stacking, your hydrogen bonding, whatever business.
So
Uh,
if I am to model this kind of interaction, so
Hmm.
normally those kind of like the ones which where transcription factors bind to the DNA, all those are modeled through Hill functions and all those things, right? I mean, yeah. So if I want to model these things, like, is there a standard way to model it, or would be different from that
because
model?
I think DNA base pairing is like from a model perspective pretty well understood, like how like all the stacking parameters, roll, pitch, yaw, whatever, like all those are pretty well studied. So I think like you can use similar parameters for this sort of structures. The only problem being that your normal DNA doesn't bulge out like this. So like you will have to run some sequence analysis to figure out what is going to be the RNA secondary structure here, and then integrate that into the model, which gives some degree of difficulty to this entire problem. So that is why like at the early stages, bioinformatics on these things was a bit tricky, because you have to figure out like how to say that this portion has like some sort of a protrusion rather than some other portion. So bioinformatics on this like was slow at first, but now with like high throughput sequencing and everything, like you have big libraries and you can sort of figure out what is the secondary structure.
Just one more question. When these microRNAs are binding to this mRNA, so is it changing its tertiary structure or something? And could that lead to repression? I mean, leading to the repression?
Yeah, there is some. So this microRNA does not bind in isolation. There is another protein complex holding it together here. So the inhibitory activity mostly comes from that protein complex, like this entire protein plus this double-stranded RNA complex, recruiting other factors which stop the ribosome from running on this. I mean, stop the ribosome from translating. So even if some secondary structure is induced, they are contributing together. It's you cannot like say that the secondary structure, induced secondary structure, is the sole contributor. There are multiple contributing factors to the inhibitory activity.
Okay, thank you.
Transcription. There are so-called splicing. You
Yes.
avoid the intron and exon region. Is it related to that or it's?
No.
I think
Because I mean, intron and exons, the cleavage happens because the DNA sequences have some specific signatures there. And then again, the intronic cleavage, which happens, is also carried out by RNA itself. So there is a other class of something called small nucleolar RNAs, SNO RNAs, which are involved in all this splicing and whatever business. But yeah, so here this is a different thing.
There is a, maybe you mentioned that there is a gene that codes for each of these, or from which these blue
Yes.
and the red are made.
Yes.
And is this existing in a normal C. elegans worm, or
Yes,
is
yes,
it only if it is mutated?
No. This is how it should be normally.
Okay.
Right? In the mutants, in one mutant, this thing is absent. The red part is
I
absent.
see.
So this protein is made when it should not be, because it's
I
not
see.
being able to repressed. In another mutant, this part, the blue part is missing. The red part is there, but now the red part has nowhere to bind.
I see.
So again, like the blue is again expressed because it's unable to be regulated.
And I don't know if it's as simple as that. If that is bound like that, then that part is not made into proteins or whatever. Is
Yes,
that?
so
Yeah.
here there is a bigger protein complex holding it in place.
Okay.
That protein complex, when it finds that there is some degree of matching like this, it will call other proteins. So it will recruit other proteins to inhibit ribosomal activity on this mRNA. There will be no translation if it's inhibitory. You can have activatory miRNAs as well, but they are very rare and like very few in number. Yes.
So, but it is not because the ribosome cannot move on this.
The ribosome has the ribosome won't be recruited at all. Like
It's not that this will block the movement.
no, because it's sitting at the end. The ribosome can already walk over. So this is at the tail end of the thing. Ribosome has, if the ribosome wanted to, and it could bind, it would have translated the entire protein before it reaches here.
Okay.
Okay. So going ahead, what was the results of their seminal work? The result was deafening silence. No one apart from the worm community cared. And people were like, oh, this is some small dumbass C. elegans worm. What does this do? It doesn't directly compare to your eukaryotic, like higher order eukaryotes. It's just some curiosity. So understandably, they were a bit dejected probably. And then what they did was, okay, fine. Maybe this specific Lin4, Lin14, which is a worm specific gene, is not generally conserved across all animals, all eukaryotes. What we will do is we will try to search for other more conserved miRNAs. So turns out that this LET7, so Rafkun in his lab, they were able to figure out that there is another one of this like Lin4, Lin14. There is LET7 and then I think Lin41 or something, 48, whatever. So again, like miRNA and protein pair. And it was around this time when your entire genome sequencing business is taking off. So there is this algorithm called BLAST, which what it can do is it can align sequences for multiple organisms if you have the gene sequences. So like Rafkun says that what they did was they were sitting and working. They got the sequencing done for this. And all they did was open BLAST in some very old primitive browser and put in the sequences for the worm. And immediately they find hits across fly and humans. So LET7 turns out very well conserved across multiple species, starting from your tiny roundworm to 2 different types of insects. And this is humans. I don't know what this one and this one are, but like conserved across multiple evolutionary species, right? So this gave some credence to their work in the sense that this is a very general form of regulation which one can have. And as soon as this was published, now all hell broke loose, kind of. Like now everyone, like as and when a fad starts, now everyone wants to do microRNA work, right? So people started publishing a lot. People started looking at multiple different animals and multiple different gene regulatory mechanisms, in the sense that they were looking at this more from a neuronal differentiation perspective. There are other ones which are like your skin differentiation basis and stuff like that. So finally,
This is at the RNA level, right? What you have shown.
this one.
Yeah.
So this is the microRNA sequence, yes.
Okay.
And then this microRNA is going, like this portion is going to go and bind its cognate mRNA, which it's going to inhibit.
So this is exactly 7 base pairs as you have shown there.
Yes.
And does it have a corresponding thing in the genome also where you can match like this? Or?
Yeah. So
and that will also be 7 base pairs, or is that something else?
So basically the 7 base pair thing here is this region. So this is something called a seed sequence. The seed sequence matches accurately
In the RNA.
in the partner RNA,
Okay.
partner mRNA, but the extra sequence here and the length of this hairpin depends on how much complementarity this portion will have to the rest of the RNA. So that is variable. So microRNAs can have an array of genes it can regulate depending on what are the like others complementary binding for other sequences.
So I don't know if I'm understanding. So I was asking, this is produced from the DNA, right?
Both
This
of them,
RNA itself.
both of them are produced from DNA.
So if
Yes.
you check in the DNA, will there be some conserved
Yeah.
sequence?
The mRNA partner is also conserved in the
Okay.
sense ki like the this
So
region.
those
in
are
the-
complementary pairs or what between
Uh,
RNA and DNA?
So there is
So I'm guess I don't know the map between an RNA and a DNA. what is the-
so from the DNA, so the DNA is double stranded. Let's say my gene is here,
Mhm.
right? So this gene is going to create an RNA here, and this RNA is going to give rise to a protein. That is why this one is called sense. That's the messenger RNA which is being made. And since it will directly be translated into protein, it's called a sense strand, sense RNA, because it makes sense to the ribosome. But now, when you are creating this microRNAs, as I will show in the biogenesis how it's made, it will have this double hairpin loop kind of a thing. And from this region, this part will match the sense. The complementary portion here matches the antisense. So this antisense portion will come and bind to the sense portion here. So that is how the microRNA recognizes the mRNA.
So microRNA and so on cannot be translated.
No. The microRNA does not code for any protein. No.
I see. That's
Yeah.
So what is is it a one-to-one relation between the DNA and the sense, RNA?
No. Whatever the DNA is, the sense will be complementary to it.
Huh.
Huh.
Clarify that.
Yeah.
The DNA is double stranded. So supposing you have an 18,
Yeah.
blah, 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 it has to be read in this direction. The complement here will look like P, A,
CG.
yeah.
Huh.
C, G, P, T, and that is read in this direction. But now the RNA polymerase reads this and makes a complementary RNA which matches this. So it'll make it A, U, G, C, A. This is basically complementary to the other strand.
Yeah.
So
Okay.
it matches the first.
And so if one wants to look at that conserved thing, one can check in the DNA also is all I
Yes.
was asking.
You
So
can.
that will also be 7 base pairs. Which
Yes.
will match across
Huh.
those.
But
species.
then you will have some variable match in this region, which can be anywhere. Like
I see.
the length of that variable region can vary, but this 7 length seed sequence will match. Yes.
That has also been looked at and matched.
Yes.
Yeah.
As always, there are some exceptions where the entire 7 doesn't match, but in most cases, this initial 7 sequence, like 7 nucleotides, are the seed sequence, and it will match.
The phenotype of this LAT7 mutant look like? Do you know?
Oh, this one I did not look at. Yeah. These are also heterochronic mutants, by the way. So like some cell type will be some other cell type in some other larval stage, but I'll have to take a look at which specific cell type this was. You probably don't want to see some deformed human baby. Anyway, so this is how it mostly works, in the sense this is the protein machinery involved in getting your miRNA silencing to work. So here, this thing, this entire hairpin loop thing is the newly synthesized, unprocessed microRNA. So this is happening inside the nucleus. So inside the nucleus, there is this protein called drosha, and then some partner protein. What this will do is it will cleave at some site to give you some structure like this. Then this structure, which is the pre-microRNA, So this pre-microRNA will now be kicked out of the nucleus by some transporter protein called exportin 5. And once it is in the nucleus, you have another set of proteins, which is called dicer, which will cleave it further to give you something of a double-stranded RNA. And this double-stranded RNA is going to be coupled to this protein called AGO, Argonaut. And this Argonaut, what it will now do is, it will remove one strand, which was the same DNA sequence there. So it keeps the mRNA, the strand which will be complementary to the mRNA, this red one, and now it's going to bind to the mRNA, which it wants to inhibit. Is this clear? So multiple sites. So this is some processing happening inside the nucleus, some processing happening in the cytoplasm, and then finally it's carrying out its activity.
So some specific class of mRNAs.
Yes, yes.
So they are there to control their activities.
So this is
Transcription
gene
activity.
regulation, but not by not by binding directly to the DNA, but rather binding to the mRNA.
mRNA. Okay.
Yes.
Thank you.
So and these other figures are showing different variations. So this is how it happens canonically. These are all non-canonical ways it can work. Just here for completion's sake, but mostly this is the pathway which it's going to take. And now activity wise, so you can see that these microRNAs, they will come and bind to the 3' region, untranslated region. And depending on the degree of strength of this binding, it can give rise to two things. It can give rise to translational repression if the binding is weak. If the binding is very strong, then it triggers RNA degradation, mRNA degradation. So the reason it does this, that the binding is strong and it wants to degrade, is that RNA interference is actually an immune response to viruses. Viruses come with double-stranded RNAs, and many viruses come with double-stranded RNAs. So the way you combat the viral infection is you take the viral DNA, chop it up, remove anything complementary to that, because basically for RNA viruses, it will be retro transcribed to DNA and then whatever, doesn't matter. So basically you want anything with the viral sequences to be gone. So that is why if there is strong binding, basically the signature of a virus, you want it to be gone. You don't care whether it's the body ka thing or not, you want the infection to be over. So that is what can happen. It can also bind to the 5' end of the untranslated region. And in some cases, like in case of this microRNA 10A and its cognate gene RPS16, it can increase translation as well. So these things sort of depend on case by case basis. And yeah, so you can have degradation, repression, all of that.
Strong binding serves a useful purpose also?
Yes,
You
because
want that going
it's
on.
clearing of viral infection.
No, not in that sense, but is it doing something useful for the functioning of the cell? And not from the viral, but is there a self, whatever belongs to the organism, is that going on also?
Generally, the miRNAs, if they are binding to their cognate partners, they are weak binding. It does not want to degrade something self unless it's an infection or something.
So in context of viral infection, you're saying this microRNAs can repress the translation of the viral mRNA, is that?
No, they're not repressing. I mean, they're repressing by removing them. Yeah, because if you bind strongly and you suspect that this is a viral RNA, you degrade anything with that viral sequence. So in the case of viruses, how this will work is it's mostly like how siRNAs work actually. So your viral RNA is this. You chop this, you take all these sequences and you load these sequences into Argonaut and scan for anything which matches this. So anything complementary, any RNA from any mRNA from the virus being translated, they will immediately be stopped and removed. So that is what you want to do. You don't want a single one of them to survive. So here the microRNAs are being made from the degradation of the viral double-stranded RNA. Right? So if the body itself is synthesizing, if the organism's DNA.
So in the C. elegans case, you have this Lin4 and Lin14.
Huh.
Both Lin4 and Lin14 are part of the native C. elegans structure.
Okay.
And Lin4 is repressing Lin14.
Yeah.
Why has it evolved to do that? What is the
Okay.
role it has within the organism itself?
So this mechanism, right? It can serve both as your regulation. It serve as a purpose, has a purpose for regulatory things, right? Gene regulation, as well as it can like be co-opted for immune response. So it is co-opted for immune response in the case of when you have strong binding and you degrade the viral RNA. But generally, like in absence of this, it's going to be regulatory. Does that answer your question to some degree?
Sequence itself is not in the native DNA, right? For
The
like
viral one,
strong,
no.
the strong thing for killing the viral thing.
No.
So how is that? Maybe it's a tangent, but where is that?
The virus also makes copies from its own genomic materials.
So it comes from there.
So it's the viral copies which you want to be immediately removed.
No, you're talking about removing the mRNA.
Removing the mRNA which is being
By
synthesized
strong
from
binding with the microRNA.
No. I mean this machinery when it is co-opted for immune response, it takes strong binding as a signature of viral infection. So when it is performing its normal regulatory role.
When you say it, which one?
The Argonaut plus microRNA thing.
Okay.
So when the Argonaut microRNA complex is performing its normal regulatory role, if it sees strong binding, then it considers it to be a signature of viral infection and then goes on to degrade it.
I see. Whereas if it is weak binding.
If it is weak binding, it will just sit and regulate the expression. It will. The sequence itself, right? The sequence itself carries the signature.
Strongly match the viral sequence.
So here the RNA being loaded, like the sort of the guide RNA being loaded, is the viral RNA itself. Because you have nucleases, endonucleases swimming around, they are cleaving the viral RNA that is being loaded into your Argonaut, and then everything is being chopped off.
Okay. Yeah.
Many of them.
Yes.
Which will do the job depending on their sequence.
Depending on which ones are being expressed, yes. So so this is more or less the biological machinery which goes into it. And these are most of the slides I have, but finally, like at the end, what I want to discuss is something of the sort key, why did evolution now choose to have two of these regulatory mechanisms? What is bad with transcription factors? I mean, transcription factors have been working for like since the earliest times, right? If bacteria have a transcription factors, it works for bacteria and they can respond to any sort of, environmental insults thrown at them. So what is the point of having a more complicated regulation like this?
Secondary help.
This is secondary, yes. So
they're only in somewhat higher organisms.
These are eukaryotic. Yes. Bacteria don't have this. Bacteria have transcription factors. Yeah. So these are like more of metazoa stages, but okay. I mean, that's classification business. Okay, but now so what people then try to do is, okay, fine. What I will do is I will take some organism, let's say C. elegans. I will remove all the microRNAs from the gene sequence because now I can do targeted deletions. If I remove all the microRNAs, does the organism develop normally? So it turns out that most of the microRNAs, if you remove, apart from the very critical ones, the organism will be fine. On the other hand, if you try to do this with transcription factors, even if you remove one transcription factor, it's going to have severe effects. So microRNAs in general have less phenotypic power in some way. But then again, we come back to the question, then why have this? It is redundancy, yes, but this one has a bit more than redundancy. Like redundancy is good because again, if some component fails, some other component comes. The thing is, let's say you want to regulate some gene via a transcription factor. And that transcription factor is not present in the cell at this moment when the signal is coming. You have to make an mRNA. That mRNA has to go into the cytosol, has to be converted into a protein, has to go back inside the nucleus, sit on its DNA partner, then regulate the gene, right? And this is going to take a whole bunch of time. On the other hand, the shorter RNA sequence is easier to synthesize, and the timescales involved for your regulation is much faster. Again, another thing is the site of activity. The miRNA is already being put into your cytosol where the protein synthesis is taking place. If you want to do anything with the transcription factors, you have to first activate the transcription factor, bind it to some now importing, like some protein which will take it inside, and then again it has to do all the business of finding its partner. So the timescales involved in your microRNA regulation is much shorter compared to the response times you will need for transcription factor based ones, provided that your transcription factor has to be synthesized first. Otherwise, they are more or less similar. Another important factor is that these microRNAs can be what you would call can be transported to specific cellular compartments. In the sense that, let's say you have some sequence, it binds to this argonaut, and then this argonaut can be loaded onto some protein, some motor protein, or some vesicle, and sent to some specific cellular compartment, and you can have very localized activity. So let's say your transcription factor, if it is inside the nucleus, it's going to bind all the places it like has binding sites to. But if you locate your regulatory factor into a single organelle, you can do regulation at a much higher pinpoint scale. A good example of this is actually the case in neuronal synapses. So it was shown that, let's say you have a neuron body here, and then you have a very long tail, and then synapses here. So you can do so that your microRNA is present, one type of microRNA is present only at this synapse. And then it will regulate that production of that protein because the ribosome is sitting here, right? Synthesis is happening here. It will regulate the transcription only for a very specific single synapse. So the degree of precision which you can create using microRNAs is much higher compared to what you can get with spatially, of course, or what you can get with transcription factors. So these are sort of the reasons why, evolution has dumped time and energy into like generating these things.
RNA stays there or it gets degraded after it's done?
So the microRNA,
Yeah.
mRNA, that
Sorry, the
entire complex.
microRNA there, you were talking about.
So the microRNAs also have a half-life. They will also get gradually degraded, but like till they persist, they will carry on the repressive activity. Yes.
So you're talking about neurotransmitters that are produced.
So
the
like yeah. So
proteins.
whatever biological
Proteins
processes going
necessary.
on at a specific synapse, you can tinker
Okay,
around
many,
with it.
many of them.
So maybe it can be synthesis of your neurotransmitter. It can be loading of the neurotransmitter into a vesicle, whatever, or fusion of synapses to your
Okay.
membrane there.
Transport the specific microRNA to that specific cell.
Yes, yes. Because you can have these argonauts have some degree of specificity, and the argonauts can also be transferred, and they can also be loaded into vesicles and then transferred. Like let's say you have some Golgi sitting here, the Golgi now shuttles some complex.
Send it to the right.
Yeah, so that can happen, like that degree of precision one can get. Yes.
Followed, is it?
Sorry.
Dell's hypothesis of, the neurons are generally either excitatory or inhibitory, not both.
I mean, no, here it is, again, here it is not, it's not as if this is an excitatory synapse and this is an inhibitory synapse. It is just regulating the synaptic activity. I'm not saying that you will have two different synapses here. Most neurons don't synthesize a plethora of neurotransmitters. They are very specific, right? So the same here.
So the strength is just modulated,
the-
but
huh,
the sign is same.
The sign is same, yes. This was a fairly recent paper. I can send it to you. No, in some cases it can be up regulating as well, rarely. No, but mostly inhibitory.
Yes.
Ah.
What is the opposite of inhibitory?
Excitatory.
Oh, excite.
Huh.
How can it play a role as an excitatory? Here it comes,
So-
sits somewhere and then
Huh.
blocks.
So again, like it
picture
depends
part.
on the partners it is recruiting. So in some cases, depending on cellular state, it can now recruit activating things.
Yes.
So very noisy systems. Yeah. And also very specific to some specific RNAs. Like in this example, this one, MIR10A. So this one increases translation. So this is increasing the amount of protein, but only if it is.
It's there, it will increase some
Huh, yes.
Binding to the 5'.
It is binding to the 5' end. That's also different. Instead of binding to the 3' end, it is at the 5'.
So it is helping to recruit the ribosome to the 5' end or something like that?
This one actually I did not go into like the exact molecular mechanism how it is doing, because like again you can see that this one it binds at the middle and then degrades. So there is a whole bunch of probably different partners it's recruiting. Yeah. Any more questions?
Is it textbook stuff now?
It
Whatever you're
is
talking-
textbook, yes.
It's
Now
very-
it is
Standard.
it is standard, yeah.
Okay.
And actually, so I was telling at the beginning that you had something called forward genetics, in the sense that you take a mutagen, throw it at an animal and pray that it hits whatever gene you want. Now you can do something called reverse genetics. So you take the animal, you know which gene you want to play around with. You put the miRNA for that gene in the animal. So now you can pinpoint what gene you want to, regulate and stuff. So this is something called reverse genetics, which is only possible because you know the sequence of that animal. If you don't know the gene sequence, then you can't do this reverse genetics thing. But so all this research, right, that went into like elucidating this entire pathway can now be used to target any gene anywhere at any time. So you have both spatiotemporal resolution for the gene you want to, target.
Who does this thing kind of thing in India?
Oh, any, so if you look at any developmental biology lab,
They
right?
do that.
So they will have, they will talk of things called knockdowns
Hmm.
or knockouts. So knockout is you have kicked an entire gene, deleted the entire gene.
Yeah.
Knockdown means that you have put some RNAi related
That's a
things,
knockdown now.
which reduces the activity of the gene. Because if you completely remove the gene, probably the animal dies. So if you see any biologists talking about knockdowns and all, they are working with this under the hood. It's all this.
This CRISPR-Cas technique, is that useful in deleting these genes or?
CRISPR-Cas will, you can use CRISPR-Cas for both insertions and deletions.
Okay.
So CRISPR, you can like completely remove whatever gene you want or the gene sequence you want from some region. But what you can also do is you can put some specific sequence in. So let's say I want to regulate some gene. For example, like actually what this. So one of the experiments, these Rafkun and Ambrose, oh my God, this is not gonna work. Yeah, it will not take much space. So what Rafkun and Ambrose wanted to show was that you have some relation like this. Your lin4 is going to repress lin14. How do you show this? So you take a gene. You first take the lin14 gene, okay? So this is the lin14 gene. We know that this lin4 is gonna come and bind to the 3' region here. So you don't change this box. Now what you do is you completely remove this box, this region, and put some other gene which will give you some visual signature. So this gene is called beta galactosidase. So if you give it galactose, you will see some fluorescence in the whatever picture you were getting. Okay. So what they created was a construct like this, and they were able to show that if you have Lin4, this beta galactosidase is present at the normal time point and then goes down as normal development will happen. If you have more Lin4, this beta galactosidase is even less expressed. If you have no Lin4, this is always expressed. So so using CRISPR-Cas, they had to do a whole bunch of cloning and recombination, whatever to do this. Using CRISPR-Cas, you can just one step, you can buy kits nowadays actually. So you don't even have to do most of the chemical work. You can just tell it to some company. They will give you this, take 1 microliter from this vial, 1 microliter from this vial, mix and inject. That is all you have to do. And that will go and whatever gene you want, it will cut and put beta gal in there. So it's the ease which these things provide you is amazing.
So is that how it is being done these days in all?
Yeah. I mean, with CRISPR, this is mostly how it works. Yeah. You just take whatever, put whatever, you're done.
Are you done with the talk?
Yeah, this is the last slide.
Okay, we have lots of questions, so thanks everyone for attending and thanks.
Yes. Thank you.
So we still have a chemistry and maybe the economics Nobels to talk about, and any volunteers, please go ahead. I mean, I checked with Vani and she was not very sure. Anyway, thanks everyone.